So here on the board above is what we've talked about so far. So for this next quiz coming up on Tuesday, it's gonna be all protozoa, everything. So everything's fair game, protozoa. The only thing you don't have to know yet is microscopy identification, because we'll do that in the lab. So everything else about protozoa, the classes, transmission, we talked about life cycles, about the cyst and trophozoite form. We'll talk a little bit more about that today. We talked about, where are we today? Go back up here. Okay, we talked about fecal oral transmission last time. Just so you know too, on the quizzes, from now on you get full points if you underline, and you get half points if you don't underline. So now it's points coming off. So underline your organisms, genus, species, from here on out. Okay, so we went back through the classes, and that was it. So it does, hopefully it will slow down just these couple of weeks while we're doing protozoa and helminths, just because we kind of get enough time like to thoroughly cover them. We get 2-3 lectures to really cover helminths, protozoa. But then as soon as we get into immunology, we're gonna pick right back up again. And as soon as we get into diagnostics, we're gonna pick up again, because those topics can be challenging. So use this to get your best grades you could possibly get ahead right now. There's not a lot of reading. So just try and power through and really maybe focus on lab more if you're feeling like you're doing really well in lecture with these topics. If you're not feeling like you're doing well with these topics, then get ahead right now so that you can focus on getting a 10 out of 10 on these next few quizzes. Okay, so I just put this on here. I just found this on the internet. I thought it was just kind of a cool way to separate. They're not totally comprehensive, but I just thought it was kind of a cool visual from the internet about bacterial versus viral versus protozoal versus fungal. So I don't think that's a bad idea for you to do something like that, or if you're a table person, for you to start comparing all the organisms as we go through. So however you want to organize it for yourself, but yeah, I just thought that was kind of cool. Okay, so we're gonna keep going with protozoa. So what I want you to know as far as life cycle is that almost all of them have the cyst and trophozoite form. I don't care necessarily that Trichomonas and Trypanosoma don't have a cyst form, just know that most of them do. That's fine. Most of them have a cyst and trophozoite form. And that's gonna come into play when we talk about diagnostics. So we're gonna talk about diagnostics today, and we'll bring transmission and life cycle back up again today. Metabolism, the only one that you do need to know is Leishmania is aerobic. Everything else is either facultative or anaerobes. You can just put them all into one category B. Category A, there's only Leishmania that's aerobic. Everything else is either facultative or anaerobes.
So you don't want us to know the difference between which ones are facultative and which ones are anaerobic?
No. You know, the reason, I think I did that last semester, but you know, are you really gonna need to know that? Like, no. In your life, in your clinical lifestyle, life cycle, you won't need to know that. So what I think that why it's important to know it's that a lot of them are facultative or anaerobes is because this is how we're gonna treat them. So we'll look at our medication, our pharmaceutical today. So transmission, you do need to know transmission, how those are transmitted, where they come from and where they are. I do think that's important to know. And then their tropic tissue, it has to do with how they're transmitted, so it makes sense. It's not something extra you need to memorize. Okay, so let's talk about repro. So repro, even though they're eukaryotic, they mostly do binary fission. And I mean, that kind of makes sense, right? Because to go through meiosis, what do you need to go through meiosis? I'm asking that in a weird way. Can somebody answer that, even though I didn't ask it in a very clear way? What do they need to undergo meiosis? Yes, they need gametes. Yes, awesome. So they need a separate sex cell. right? You need a separate sperm or egg where they're only one cell. So it kind of makes sense, right? There's only one cell. They don't have another cell that can undergo meiosis. They only have one, where we have trillions. So we have separate cells that can undergo meiosis, so that we can reproduce, so that we can have that cell with half the number of chromosomes, so that they can meet up. Well, eukaryotes, these protozoa especially, don't have a separate cell. So
Because they're unicellular?
because they're unicellular.
Gotcha.
Awesome, yes. So mostly binary fission is how they reproduce. They will go through the cell cycle because they don't, not like bacteria that don't really go through the cell cycle, because bacteria just reproduce, binary fission. They're gonna just copy, split. Well, eukaryotes or protozoa have more timed, and they'll have like a quiescent stage, which we didn't really talk about, G0, but they don't reproduce at such a rapid rate. So they do go through organelle stage, they go through the synthesis stage, they go through the splitting stage, all that kind of stuff. So it's a good time to review right here. Oh, here I put here, mostly asexual, some are sexual, but we'll talk about it. Review mitosis, and there's one little example of meiosis, but it's not really meiosis, so I'll just kind of keep it simple if we can. So here's this process of binary fission. We already know what that looks like. It's just an image I found on the internet that I thought was kind of cool. So time to go back over the cell cycle, know the interphase, mitosis, cytokinesis. So here are the chromosomes lining up on the metaphase plate, counting chromosomes. It's a good time to review that. So let's talk about sexual reproduction, but I'm just going to give you one little tiny example. So they can undergo sexual reproduction, and again, we worry about sexual reproduction because that's how drug resistance can be acquired so easily, is through genetic variation. So what they can do is sometimes their nuclei, they'll go through the stage of synthesis through S phase and interphase, and they'll reproduce, they'll replicate their genome, and then part of that nucleus will split off, and then it'll replicate its genome again. So it's almost like it created a little egg. So will create these little things called micronuclei. And I don't care that you understand how that happens, just know that they do sexually reproduce, that can happen. And they can share those little micronuclei between protozoa, so that sexual reproduction will lead to drug resistance and genetic variation in those organisms. So it can happen, sexual reproduction, and it's through micronuclei, but I don't care that you know that. So just so you know, it's possible. So this is just a meiosis review of what that might look like. It's just a good time to review it. Always just keep it in the front of your memory. There was a little spiel I wanted to do today. What was it? Oh, I did want to mention something. So you know how sometimes when we get here early, when we have something to memorize, like rote memorization, when I make you write something and on the board and we erase it and we write it again, erase it, write it again. So in my other class, anatomy and physiology, we did that for, we just did the endocrine organs and what hormones they secrete and what actions those have in the body. So we had a little chart, endocrine organs, hormones, responses, and we had a chart. So I passed out these yellow pages and I had them fold them in fours, and we wrote it and then flipped it over, wrote it again. I hit it, and then they tried to do it 2 or 3 times on their own. And almost everyone got a 10 out of 10 on that quiz. I was like, what just happened? And they were like, oh my gosh, we got so much out of that. So even though I've said repeat, I don't know if anybody goes through that process of like hand writing it, or writing it if you have a pen and an iPad. Typing doesn't work as well. Handwriting something, getting rid of it, handwriting it again. Get rid of it, handwrite it again. That really does help. So that was my little spiel for the day is, if you're gonna know the 7 F's, then write them down, hide it from yourself, see how many you can guess. Try it again, try it again, try it again. And I do that for all my classes. That's how I got A's in chemistry, organic chemistry, except my last class where I got a 38 out of 100, it ended up being a B. In my calculus and all the classes that I've taken, because what I do is I'll just write it and toss it, and write it again, and toss it, and write it again, and toss it. And it's just that memorization of doing it over and over is such a great tactic for me anyway, so try it. Okay, so going through meiosis or going through mitosis, writing those out, erasing it, write it again. Okay, diagnostic tools. So we're gonna watch a couple of videos because I think this is a really cool way for you to learn about protozoa and kind of what we're looking for. So this is our first video that we're gonna watch. And this is an ova and parasite exam. So if you're working in a clinic, or so you might, if you end up working in a clinic, or you end up working in a hospital, like in a lab or pathology lab, but sometimes clinics will do it and you don't have to run it through the hospital. So my sister is a physician. She works at a hospital. She worked for Kaiser for a while, and then she worked for Carbon Health Clinic for a while, and now she's only working at Catalina Hospital on Catalina Island off LA, because it's considered a rural hospital and they're helping her pay back her loans. So she works there sometimes 2 days a week or 3 days a week, and because it's a smaller hospital, she does all this herself. So she'll either do it herself, or her nurse practitioner will do it, or her medical assistant will do it, or a nurse in the office will do it, or she'll do it herself depending on how busy everybody is. So, any kind of clinician, you might end up doing this stuff yourself. So I think it's cool to know. And also I think it's cool to know when you send someone's stool sample or blood sample or sputum sample, what they're doing with it. What do they do with it, and what results are you looking for? Okay, so I'm going to play this one.
How to do stool routine examination. To do stool routine examination and to making a smear, we need normal saline. This normal saline is easily available from pharmaceutical companies, or you can take this from your nursing department and can put in small bottle with dropper. That is more comfortable and easy to use. Now you see, NSS means normal saline solution. For this stool analysis routine examination, we need stool in sterile container, and we need one glass slide and one cover slip. First, when we will put one drop of normal saline on the one side of the slide or in the center as you wish, and then when you will open the bottle, first you have to see the color of the stool, and when you will make the smear either with the stick in the container, or you can use any dropper. With the help of that, we will see the consistency. Colors means if it is brown, yellow, green, whatever color you will mention in your report, and consistency means it was formed, semi-formed, loose, soft, mucoid, watery. So whatever the consistency will be. So we will mix with in the normal saline and the sample, and we will cover with the cover slip. Now we will set the slide in the microscope. First you have to use 10x lens and keep on the light of the microscope, and with the help of coarse adjustment we move the stage full downside and then slowly bring it up. Now I will show you. As you can see, I am moving the coarse adjustment and making the stage up, and at one point I will stop where I will see the focus is clear for 10x. I will show you also, and then I will change to 40x. We have to report stool analysis at 40x. Now this is the 10x view. In this 10x, just we have to make our focus clear, and then I will change the lens to 40x. In stool analysis, now it is 40x. Now you can see there is downside, there is giardia cyst, and upper also there is one giardia cyst. In stool analysis microscopy, we will search for parasites, we will search for bacteria, yeast cell, undigested food, pus cells, RBCs, red cells. These all things we will search. Starch, fat globules. These all things we will search in stool microscopy. And there are routine parasites like Entamoeba, histolytic Entamoeba, coli, giardia, lambia. These are the normal routine parasites that you will observe. Other than also there is like Ascaris, Antrobius, H. nana. There are different parasites. I have already shared microscopic images. That all we will report. So in reporting we will report color, consistency, pus cells, red cells, if there is any parasite, it is in cyst form or trophozoite form, and then bacteria, and if there is undigested food, and there is yeast cell or fat globules, that we will report. And in corpus cells and red cells we will report like 1 to 2, 0 to 2 according to the field. We will take average and we will report. And parasites and bacteria and other things we will report like +1, +2, occasional, rare, like that we will report. So this is the stool analysis.
Okay. So I thought that was cool because she explained what she's looking for. So here's a cyst right here. So so I am going to try to ask the question. Let me see how I can ask it. So ova and parasite. So ova, what it means is eggs. So those are eggs of helminths, which we haven't talked about yet. So she mentioned in there a couple of different helminths that we're going to review. So they'll have eggs in the stool. So when you're looking for protozoa, why are we looking for cysts? Yeah.
I want you to a wild guess.
Yeah.
Because the cysts are what we're gonna be pooping out, right? And then the live form is going to be inside of us, and then the cyst form is what we're looking at.
Exactly. So if you were looking for trophozoites and you didn't find any, you would be looking for the wrong thing, because they'll insist on the way out because it's not the environment that they need. They need a warm environment, they need the correct acidity, they need the correct nutrients to be in the trophozoite form. In the stool, you might find a trophozoite or two and they'll probably be dead, but you'll find cysts generally if your patient has a parasitic infection. So that's what you're looking for, are cysts generally of any of these ones. Let me get my little pointer. So here, we'd be looking for cyst, 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 and that's it for all of our fecal orals. So that's what a stool sample looks like. She also mentioned a couple things that I thought was interesting too, is that you might look for white blood cells. If there's any sort of infection in the tissue, your body's going to be making more white blood cells, so there might be white blood cells in the stool. There might be red blood cells in the stool. If there's red blood cells in the stool, it's a sign of dysentery, so maybe. But one thing too I wanted you to keep in mind is that with our class, how we think about things, we think from the micro perspective. But if your patient has some sort of gastroenteritis, it might be diet related. It might not be a parasite or a bacteria or a virus or anything at all. It might be because they had 2 cheeseburgers for breakfast and now their stomach hurts, and you're like, oh, you know. So just keep that in the back of your mind. Okay, so I know it seems gross, and it seems gross to do a stool sample, but if you are going into the healthcare field, this might be your life. You'll get used to it. I don't want to tell you. Should I tell you? Should I not tell you?
Should tell us.
All right, I'll tell you. So my sister that went through medical school, she you have to have all of these hours of actual hands-on clinical work. So as her part of their training, they go through and there's women's rooms and men's rooms, and they go through and do a vaginal swab but the whole medical school goes through and does a vaginal swab for the same people sit in the same room. And then they'll do like prostate exams, and so they do all of urogenital, and they just sit in there. They're the like model patient for the people who run through the whole gamut of medical students. So pretty gnarly, but yes, you will have to do this if you're going into the clinical setting, but it does get easier. I think I would say like my husband gets more, he definitely has a higher tolerance for things than I do, but it's just experience too, and eventually you'll be like, oh, I've done 15 catheters this week, I'm fine with it. You'll get there, but it takes a little bit of time in the beginning. Okay, so here's what we're looking for when you think your patient might have an STI. So if you think your patient might have an STI, they'll probably do a blood test first. You might always do a blood test first, so depending on what you think it might be, you'll run a CBC. It's called a complete blood count, and we'll go over what that means and what you're looking for in a CBC. But they'll take a blood sample first to kind of see, like, is it looking more infectious? Do they have a high white blood cell count? Is there something else going on? So they'll start there. But if you think your patient has an STI, they'll do a penile or a vaginal swab. So here is, it doesn't show it obviously, but it shows what the results are going to look like. So that's trick. Oh, I don't think this one talks. So that's when they literally take a Q-tip and do a vaginal swab, or they'll stick it up the urethra of the penis and do a penile swab. And this is when you do a wet mount onto a slide, that's what trick will look like. So I'm not gonna have you memorize this, that the trick doesn't have a cyst form, so they're always gonna be this live trophozoite form. You don't have to memorize that, but it would make sense logically that you wouldn't find a cyst form because you're taking it directly from the area where the trophozoite lives, right? So you don't have to memorize that. You just know if it's a penile swab, that's going to be the trophozoite or a vaginal swab because that's where the organism is living and thriving, right? Okay. Yeah.
And from the time you take it out to the time you look at it on the thing, it's not going to die?
They might be dead on there, and you'll be able to see them. But you might have a couple living ones. Yeah, they'll probably still be living, I would guess. Yeah, you might have a couple of dead ones, but I bet.
But I mean, they wouldn't like insist.
They wouldn't. Well, these ones don't anyway.
Oh,
but
okay. But I mean, like another variety.
Yeah, no, yeah. They'll be in the trophozoite form so. For example, if you were, instead of to take a stool sample, if they did like a colonoscopy for a patient who had some sort of intestinal infection and they had a sample from the colon, you'd find the trophozoites there. Yeah. Okay, so for our last one, a blood smear. So this one is really cool, and for a couple reasons. I'll let you watch this one. I think it's interesting. This one's a little bit longer, but I'll cut it short towards the end. And I think it's really neat because, I mean, maybe someday I'll do it.
In order to perform a proper thick and thin blood smear, you need the following material. Gloves as universal precaution, dry and clean piece of gauze, a spreader, clean and clear glass slide of 25 by 76 millimeter approximately, small cotton balls soaked in alcohol, sterile blood lancet, a bin to discard used blood lancet, and a slide folder to protect slide from dust and the spreader, you need a slide with clean, frosted and polished edge. When you pass your finger on the edge, it should feel very smooth. If you feel any asperities or surface roughness, you should replace the spreader. Health workers should be comfortably seated with enough bench space and proper lighting. For pricking the finger, choose the middle or the ring finger. Always prick on either side of the fingertip, not in the middle. This is because the middle of the fingertip has more nerve endings than the sides. Therefore, pricking on the side is less painful for the patient. The patient should be seated comfortably. Then record a request form and a slide. Here it is done with a sticker, but it can be done on the thin smear with a pencil. Record should have at least a code or a name and the date. Check the slide is clean, free of dust or fingerprints. If not, clean it with a dry piece of gauze. Note here that the slide can be manipulated as long as you keep your finger on the edge of the slide. Massage fingertip to increase blood flow. Clean the fingertip thoroughly with a cotton ball soaked in alcohol. This step needs to be done properly in order to avoid contamination of the blood taken or infection for the patient. Open the protective wrapper of the sterile blood lancet from the back side. If you notice the protective wrapper has already been teared off or damaged, discard the blood lancet as it is no longer sterile and use a new one. Pull the lancet out of the protective wrapper and prick the side of the fingertip with a quick hand rotation. Discard the used blood lancet in a proper bin. Press slightly the finger. The blood should come out easily. If not, restart by cleaning and pricking another finger. With a dry and clean piece of gauze, wipe out the remaining alcohol and the first drop of blood as it may contain skin debris. Press the finger to have a blood drop out. Come with the slide to make contact with the top of the blood drop, but not with the finger. This is crucial if you want to take a standard blood quantity and have thick blood smear with a standard thickness. Repeat this in order to obtain 2 to 3 drops of blood for the thick smear. If the drops are small, take 3. If they are a bit bigger, take 2. The volume of blood for the thick smear should be about 6 to 10 microliter. For the thin smear, take 1 small drop, 2 to 3 microliter, about 1 centimeter away from the thick smear and in the middle of the slide. Give back the cotton ball to the patient, telling him to press on the puncture site for 1 minute. Hold the slide between your thumb and both your middle and index finger. With the other hand, take the spreader and put it in contact with the slide in front of the thin smear drop. Move the spreader backward to make contact with the blood drop. Wait for the blood to run along the spreader's edge. You can help that process by opening and closing the angle of the spreader in quick succession. Keeping an angle of approximately 45 degrees, move the spreader forward with a regular move. There is no need to apply too much pressure on the spreader, but it should stay in even contact with the slide surface when the blood is being spread. The forward move should be regular, not too fast, and completed until the spreader reaches your thumb. Then put the slide on the bench. With the corner of the spreader, join the 2 or 3 drops for the thick blood film with few circular movements in order to obtain a cycle of about 12 mm in diameter.
Okay, so what they
Place
did
the
is
slide
do
in
a
a slide
thick and
folder
thin
to
blood
protect
smear.
it from dust and flies.
Yeah.
Leave it.
So they did a thick and thin blood smear, and they are looking for, when it's under the microscope, it'll have all of the erythrocytes. And a malaria positive patient, sometimes they'll say it like looks like a happy face, or sometimes it'll have the trophozoite, or it'll have some cysts and trophozoites in the red blood cells. Sometimes it's just one red blood cell or two red blood cells, or, you know, there won't be very many that are positive from that thick and thin blood smear. But that is how they diagnose malaria. And so what I thought would be, what I think is cool about that video and why I tried to pick these videos specifically is because how medicine is performed in other countries. So I don't know if you recognize that or know that, like, this is what it's like. They don't, you know, they are working with rudimentary tools. The person that's in charge is doing every exam, so he probably did 100 that day or something like that. So it's a lot. One thing I noticed when I was in Africa, and so I was in charge of triage. So, you know, the patient would come up and I'd say, okay, do you, you know, have any symptoms right now? What are your symptoms? How are you feeling? And so sometimes that we would ask them, like, oh, I've been really tired. Do you have malaria? Do you know if you have HIV? Okay, so let me write it down on this piece of paper. Then we would weigh them, then we would measure how tall they are, ask if they have any children, have they eaten, that kind of thing. So we did this whole little triage, and then you put their piece of paper that I hand wrote with this little pen on this dirty table outdoors and on the dirty floor, and we put it in a box. And there's just papers and papers and papers in boxes. And there's no organizational system. They have no, like, digital record. Sometimes it's very difficult to understand their name or how to spell their name or what their names are. Sometimes people don't know how old they are. You know, I think that's one little thing I wanted to mention also about bedside manner is, like, the nurse sitting next to me, she was a nurse, but she was just triaging with me, and the lady comes up and says, I have a bad spirit. My spirit is bad. And she would say, you know, something is wrong with my spirit. And the nurse next to me, she is from South Carolina, and she was so annoyed. She's like, oh, your spirit is sick, and, like, so annoyed with her. And I was like, that's her belief system, you know, she thinks her spirit is, that's truly what she thinks. And being able to speak to her of, like, you know, what does that mean when your spirit is sick? How are you feeling? Like, what does that mean to you? And getting more information from her, because it doesn't help her, which is your goal as a practitioner, right? Your goal is to help your patient and understanding where they're coming from. So, yeah, so I think I thought that was cool, is to learn how they look, how they make a slide, how they make a blood smear when they're looking for bloodborne diseases, and then also just the skills and things like that you might need if you ever do clinical work outside of this country. I think it's important to know. It was mind-blowing for me. I've traveled a lot, but it was completely mind-blowing to me, the record-keeping system, just because of how crazy our record-keeping system is. Double, triple check, everything's in the computer. What is your name? What is your date of birth? Here is your thing, let me double check that. Everything's in the computer, let's do, and they were just like, what's your name? Sure, throw it in a box. Okay, that sounds pretty good. Oh, you have HIV? Throw it in the box. Like, it's pretty crazy how the healthcare system works in other countries, so I think that's just interesting to keep your mind around if you ever do work like that. Yeah.
My husband was in Argentina for a couple years, and one of the stories he tells is he would go to the doctor, and the doctor would say, look for a blackberry that's about this big, and smear it on and add some honey, and like those were the diagnosis, or those were the treatments they would get, is because they were just out in the sticks.
Right. Yeah, that's cool. I think it's cool. I think just, like, learning what other people do and trying not to judge them for what they do. And, you know, who's to say ours is the best? We have a super high hospital mortality rate. We have a really high infectious disease rate that runs between hospitals. And, you know, who's to say that because we all, you know, have all these cleaning methods and lights and floors and gurneys and all the things that we have, you know, that it's the best way. So I think it's kind of cool to be able to see what other countries do. Okay. So those were our 3 types of diagnostic tools, the ova and parasite. So remember, ovas are eggs of helminths. Parasites will be looking for, if you're looking at a stool sample, then what parasite you're looking for is the cyst form of a protozoa. If you do a swab, a penile or vaginal swab, you're looking for the trophozoite of trichomonas is what we're generally looking for, unless you find yeast, which is common, a candida infection, or if it's not yeast, if it's not a protozoa, and if it's not yeast, then remember what those cells look like. What were these cells called? When you see these, well, these are human cells. So in the vaginal swab, you got these little epithelial cells from the vaginal lining. Do you remember what those little fuzzy cells are called? Clue cells. Yes, awesome. Good job, Dr. Ferguson. So these are clue cells. So clue cells, because when you look at the epithelial cell and it's fuzzy around the outside, and then you zoom way, way in, because remember, we're zoomed out for all of this. We don't need to zoom in. But here we're gonna zoom in and then we'll find Gardnerella vaginalis. That's for BV. So it's a good idea also to look through, like, what are your STIs, what are your respiratory, what are your gastrointestinal, and we'll kind of go back over that towards the end. Yeah?
Sorry, what did you say clue cells are?
Clue cells are Gardnerella vaginalis, and that means your patient has bacterial vaginosis. Do you remember that one from the note cards? Yeah, awesome.
So they're like, they only exist when the patient has BV?
Yes.
Okay.
Yes, awesome. Because the fuzziness is the bacteria. Because you don't usually zoom in, and they don't usually use oil very often. But you'll be like, what is wrong with that epithelial cell? Why does it look so weird? Oh, it's fuzzy around the outside because it's covered in bacteria. So let's zoom in. Oh, that's Gardnerella vaginalis. They have a bacterial infection, but it's not sexually transmitted. It's a pH problem, so they'll do some sort of treatment. They'll give the patient medication, that kind of thing. Cool. Okay, so let's go through some of our diagnostics. So the diagnostics, it depends on what we're talking about, right? So it depends on if your patient comes in with a gastrointestinal infection. It depends if your patient comes in with genital issues. If your patient has insect-borne, I'll tell you what the symptoms are that. But you're gonna look at what your patient's symptoms are. So you start with looking at your patient's symptoms. And so for insect-borne, for example, so fecal-oral, they'll have gastrointestinal symptoms, right? They'll have diarrhea, maybe they've lost weight. Sometimes they'll have dysentery, things like that. So, hold on, let me go back to that really quick. I wanted to, I don't know why it always does something funky when I watch a video. There we go. Okay. So it depends on what the symptoms are. So an insect-borne, so here are the trypanosomas. Let's forget about leishmania for a second, but here, insect. These are all insect-borne, and these are all blood issues except leishmania. And so when it affects your patient's bloodstream, then your patient is having problems getting oxygen to their brain generally, because the oxygen is not you're not getting enough oxygen and glucose to all the tissues. So it's lysing red blood cells, and so your patient will feel tired, and it'll be a long-term tired. Sometimes they'll have a fever because it's systemic, it's all over their body, their whole system is called systemic. So they'll have some symptoms of systemic issues like headaches, fever, malaise, things like that. They can have chills, sometimes they get like flu-like symptoms. So then you're looking at what, depending on what the protozoa is what we're giving us our first idea about what our patient might have. And then we're gonna take a history and physical. So then we're like, okay, where have they been? What have they been doing? Where, sometimes if your patient has a bacterial or viral infection, they have had to come into contact with somebody, but we don't dig that deep because we know, like, oh, you got a bacterial infection because somebody that you ran into at the grocery store, So, but these are not as common. So you had to have gotten it from somewhere specific. So did you get it from, what, have you been drinking any weird water? Have you been traveling? That's always a huge question. Have you been anywhere traveling? If it's sexually transmitted, have you had new sex partners, multiple sex partners? Have you had unsafe sex? For insect bites, or have you been traveling? Have you noticed that you've had mosquito bites recently, or any other, like the tsetse fly or the kissing bug? Like, have you noticed an insect bite? So as far as protozoa are concerned, you'll have to dig a little bit deeper. Where have they been? And then for a protozoal infection, really, you kind of rule everything else out. You don't first think protozoa. What is that? It's like Occam's razor or something. You don't first think zebras. You first think horses. It's probably bacterial, it's probably viral. And then you're like, well, it's gone on a long time and they have a chronic cough. Well, maybe it's fungal. It's gone on a long time and they have gastroenteritis for 3 months and they went to Mexico 3 months ago. Maybe it is protozoa. Maybe it's not bacterial anymore. Maybe there's something going on. So, protozoal infections in this country are not super common. The one that's the most common is Cryptosporidium, but they probably would know where they got that. Do you swim in the local swimming hole? Do you swim in dirty swimming pools? Have you, something like that. So you're gonna ask them questions, and then you can track outbreaks. So a lot of these, you'll know because the CDC will report, and so as a clinician, you'll look up, like, is there an outbreak of crypto? Is there an outbreak of malaria? Yeah.
What is TB?
Trichomonas vaginalis.
Okay.
Did I wrote TB on there?
Yeah, you just wrote TB. Oh, on the slide.
Oh yeah. For here, oh yeah, except Trichomonas vaginalis, except TB, yeah. So, yes, because trick is more common. So, trick, it could be that. But, like, protozoa, giardia is not as common, malaria is not very common, all of the other ones are not very common. Okay, so that's kind of some of the diagnostics we might think about. So it's a good idea as we're moving along to separate out bacterial, viral, protozoa, and we move on to helminths. Okay, so treatment. So now, why are protozoa so hard to treat? We've asked this question multiple times. Yeah, they're so much like us, right? It's basically one of our cells split off and doing its own thing. So they're very hard to treat. But there's one difference that's enough of a difference for us to treat in protozoa is they're anaerobes or facultative. So that's where that topic comes back up, that most of them are anaerobic or facultative. So they have this metabolism where we can target an enzyme from their metabolism. So we're going to target an enzyme. I'll show you where this enzyme comes up. It's called pyruvate ferredoxin oxidoreductase, but you don't have to memorize that. I'll tell you where that comes in. But what we're going to target with, it's called metronidazole. Metronidazole. So this metronidazole is an anti-parasitic. Oh, I want you to remember the name Flagyl as well, because it'll be called either or.
Is it
I've
a
seen
brand name or
Flagyl
something?
A lot. I don't think it's a brand. I think it's it's like a common name for it. I don't know. I don't know why they call it. I've seen it both in literature, so I'm not 100% sure why. So flagyl or metronidazole is generally what is used as an antiparasitic. So we only have one. We don't have to remember a whole list, so that's awesome. You have to remember one and what it does. So what it does is, here's aerobic organisms, here's anaerobic organisms or facultative. So this pyruvate ferredoxin oxidoreductase is an enzyme that will break up pyruvate into acetyl-CoA in aerobic or facultative organisms. So it's going to target this enzyme. So what's going to be the problem with this protozoa? They're not going to have what? ATP. Awesome. So they're not going to be able to make ATP, so they'll die from not enough ATP to be able to run their body programs, their body systems. Okay, so
You said it targets the pyruvate?
yeah, it targets this enzyme that breaks up pyruvate into acetyl-CoA. Yeah, it targets that enzyme. Yeah.
Are you gonna memorize any of this?
No, the only thing I want you to know is it targets an enzyme in anaerobic or facultative protozoa. Does that make sense? Everybody with me on that one? Okay. So, what else? So what organism, what protozoa is this not gonna work on?
The Cruzy or the Cruciate?
Not these ones.
Leishmania.
Yep, Leishmania. So Leishmania was our only aerobic. So it doesn't work on Leishmania. You have to use something else. I don't know what they use on Leishmania actually. Probably topical. But everything else we can use metronidazole. They use other things too, sometimes other pharmaceuticals, but that's the most common. Yeah.
If they cover it, does that like get rid of the oxygen enough?
Yeah, so
Maybe.
sometimes that helps even with fungi because it's aerobic. So
So you put
it
a
will
nice
help
coating
to
of
help.
Vaseline on top of
If you
it.
keep coating it, it will help. That does help. But you know, you can't like sleep and it'll wipe it off, or shower and it'll wipe it off. But you know, it does help with fungal infections as well. Is just to coat it so that it doesn't have access to oxygen. Okay. So protozoal diseases. So you guys already know these. This is nothing else you have to memorize, but here's our ciliates, amoeboids, flagellates, and non-modal. Only drink water that's clean as F. And non-toxic. So that's my mnemonic for this time. You should feel pretty comfortable with these by now. I put some little like GTTL if that helps you. Crypto, cyclo, we got rid of cysto because it's not very common, toxo, and plasma. Okay, so protozoa wrap up and we're all done. So respiration, respiration some are aerobic, but mostly the aerobic protozoa are non-parasitic. So the only parasitic protozoa that we know of that we've talked about is Leishmania. But don't forget, there are so many protozoa out there. So many. Most of those are aerobic, but they're non-pathogenic, so we didn't study them. Some are photosynthetic, we don't study those either. Facultatives and anaerobes we talked about. A lot of them like to live in aqueous environments, so they need fluid to move around in. They're more limited than bacteria, archaea, viruses. I wouldn't say fungi maybe. Fungi are pretty limited too. They need another form to go from host to vector. That's the cyst form. Trophozoite cyst, motility, repro, we talked about that, and that was it. Okay, so we are going to, I was going to start on helminths today. Let me just introduce helminths, even though I'm not going to ask it on the next quiz, because I'm just going to give you an idea of what helminths look like, and then we're going to do our note part for the day. So we're going to, so we moved from bacteria. We talked a ton about bacteria, all of their life cycles, repro, genetics, everything, all of the antibiotics, antibiotic resistance. We talked about mutations, plasmids, horizontal gene transfer, all of that kind of stuff. Then we moved on to archaea, which we spent a hot minute on because they're non-pathogenic. The only reason we brought up archaea is because they contribute to climate change, and they're part of our tree. I think it's important just to mention. And then we are spending the whole second half of the semester on eukaryotes. So on our eukaryotes, we started talking about fungal organisms, which are molds and yeast. Molds and yeast, they're everywhere, they're ubiquitous, but most people with a competent immune system are fine. Some people that are immunocompromised have issues, and they're highly susceptible to fungal infections. Both molds and yeast they're susceptible to. What else? They have chitin, nanoproteins, glucans. Molds are aerobic, yeasts are facultative. We talked about our antifungals. We have a handful of antifungals that we had to know. And yeah, that was kind of it. And then we talked about protozoa, so we talked a lot about our protozoa. We talked about transmission and how common they are or uncommon they are. We talked about their life cycle, their metabolism, their cytopathic effects. We talked more about fecal-oral transmission. That's really appropriate with every organism except fungi, really. So helminths especially, we're going to talk a lot about fecal-oral transmission. We talked a little bit about repro, a little bit about diagnostic tools that we use. We're going to use the same diagnostic tools, well, not blood or STI, but the ova and parasite, we use that also for helminths because they are mostly fecal-orally transmitted and mostly gastrointestinal, not all. Okay, so we're here in this branch of animals. So now we're moving from tiny to bigger, bigger, bigger. Now, eukaryotes, the protozoa that are single unicellular pathogens is what we focused on in the prokaryotic realm. Now we're moving up to multicellularity. So helminths are worms. They have they have reproductive organs. They have a gastrointestinal system. They have a rudimentary nervous system. They have skin cells. Now we're moving up in complexity where they are multicellular and are able to do more, I guess I'm trying to say, than protozoa. Okay, so they are animals. So we're going to memorize this next time. We don't have to know it for this test right now, but we're gonna memorize what flatworms are and what roundworms are. There's two types of flatworms and one type of roundworms that we're like going into branches or classes. It's real gross, so just warning, it gets pretty disgusting when we start talking about helminths. But just so you know, too, they're very common, and they're common in this country, actually. There was one of my students a couple of semesters ago did this whole part of they didn't do a poster project, they did a research project, but part of their research project was people that are using tapeworms as a diet, as a way to lose weight. So, and it actually was kind of going around, I don't know, I haven't heard about it in the last year or so, but it was going around that it was a popular method to lose weight. So it actually was common here, was like going around in areas of like Los Angeles and things like that. And well, I'll talk about how incredibly dangerous that is for the patient, but also for everyone that you come in contact with, because they are you can spread them very easily. They will get through a population very rapidly. So, kids get pinworms. We'll talk more about kids will get these frequently. Animals have these. Animals have worms, and kids can get them from animals or other kids. So we'll talk more about that. So why do helminths belong in micro? Well, I'm not really sure why. I think micro is directed towards clinical medicine. Not always. Some universities or classes that offer microbiology will teach it from a more environmental perspective and teach less pathogenic and more, but we teach at this campus according to the book, and the book is a little more clinical, and I teach even probably more clinical than some of the other professors, because I think that that's how it's going to impact your lives the most. If you ever run into a protozoa, it'll be because of your health, your patient's health, your pet's health, your children's health, something like that. So I think it's important to talk about. So helminths, they are common, more common than you would ever want to know probably. They're called soil transmitted helminths. In the literature, you'll see STH, that term, a lot, but they're not all soil transmitted, so I don't really like to say that, but they're just soil transmitted helminths. They say STH when they talk about helminths all the time. And they are worms, but they are human parasitic worms. So they're not worms that are, just earthworms and things like that. Helminths are human parasites. And then also their eggs are microscopic, so that might be another reason why they lump them into our microbiology realm as well. Yeah?
Is it 25 meters a typo, or is it?
No, that is right.
Ooh.
Yes, yeah. So tapeworms can be like a quarter of a football field long. They're can be humongous. And then they just coil in people's intestines, and they can live there for a long time. Or in animals. So animals have a problem with helminths as well, and we'll talk about that. So they do have eggs. They have another stage called larvae, which we'll talk about. And then the adults are generally, you can see without a microscope, but that's just most of the ones that we'll talk about. Okay, so they're multicellular. 82 feet is the longest, I think. I don't know if it's the longest on record, but that's just generally. So we'll talk more about multicellularity next time, and we'll talk more about what cestodes are. These are tapeworms. Trematodes, these are flukes. Nematodes these are a lot we're going to talk about. Ascaris, pinworms, whipworms, hookworms, threadworms. We'll talk all about those. We'll talk about the differences between these and how to recognize them under a microscope. Okay, and then we'll talk a ton about transmission, how people get these, and how common they are. Okay, so let's do our note card, and we will head down to the lab, because we have a big lab today. We haven't done giardia yet, right?
Can you use the protozoa medication
For other anaerobes, I don't know if they use metronidazole for like anaerobic bacteria or things like that. I don't know if I have any more note cards.
I just brought a ton.
Oh yeah, sorry, I don't have any more note cards. If somebody needs to borrow one, I'll bring a bunch next time.
Something you might find
Thinking
really
someone-
interesting is,
Huh.
dogs can contract heartworm. Are you aware of that?
Yes.
And when they get really bad, you can kill them by treating it. So they have to non-surgically cut the jugular vein and pull the worms from inside their heart.
That is
pretty gross.
awful. Have you seen that before?
I've talked about it, it was like a 3 AM discussion with the doctor, but I've never seen it happen.
That is awful. I would not, I don't know if I'd recover from that. I'd have nightmares. Oh, worms gross me out so bad. Okay, so this is Giardia lamblia. We've talked a lot about it, so that is helpful. We already are pretty comfortable with it. So this is the trophozoite. This is the trophozoite if you look at it sideways because it's flat. And then here's the cyst. That's a nicer way to look at it. I like it. Let's just pretend. Okay. You guys ready to move on? Okay, this is what they look like under the microscope. These two nuclei make them look like they have little eyes. That's kind of characteristic of Giardia. So we know that, so giardiasis, that's the disease from Giardia, giardiasis. They have flagella, cysts or fecal oral. They can be mechanical. They have to be mechanical, do they? They're not biologically. Yeah, no biological transmission. The trophozoite will live in the gastrointestinal system. Diarrhea, gas. One common thing from people who have Giardia is greasy. Their feces look greasy, and that's because everything they're eating is not being digested at all. So it's coming out more the way it came in. But Giardia will more uncharacteristically of other gastrointestinal infections is they almost like coat the whole colon. So it's almost like the colon doesn't even exist. Stomach cramps, pain, nausea, dehydration, malnutrition, mortality. Common hikers, travelers, 1 million per year in the United States. That seems crazy high to me, but 50 to 70% of people that live in India have Giardia. 3/4 of the population. 200 million worldwide, 500,000 deaths worldwide from Giardia. My mom literally almost died. She did. She was like 80 pounds. She's little anyway. So she was so sick she could not eat. That's it.
Is that 50 to 70% in India, the number of people that are living with it actively?
That live with it. Is that, and that's like a wild estimate because nobody really knows, so they guess. Yeah. It's rampant there. So if you do travel to India, eat cooked fruits and vegetables, not fruit, because you can't really cook fruit, but cooked vegetables, well cooked meat, and then just anything, just washing it constantly. Yeah. Yeah, very good question. So their bodies, they can live with it. So because they probably acquire it after they're born, pretty rapidly after they're born, and then their bodies will just deal with it. And so a lot of people will be underweight or malnutrition, but they can live with it their whole life. Yeah.
So if you live in India and you're around how people have it, how do those people not get it? How do like the small people?
So the people don't get it is because, so here, you can live in the same house with somebody with Giardia even here, because it's fecal orally transmitted. So as long as you're really careful that you don't transmit, like washing hands, washing the toilet,